All right, what's going on, guys? Welcome back to the channel. We got another breakdown coming at you guys. This is passage number 3. This is a BB passage, okay? And I'm going to show you guys where to highlight, how to make all these details make sense, okay? How to interpret these figures. I'm going to show you guys how to pick the best answer. I'm going to show you guys everything that you need to know to increase your target score. My bad, to increase the points that you get on the MCAT, okay? To get more questions right. Now, before I do this and break this down, make sure you read the passage on your own first and answer these questions on your own first. So I'm going to scroll down, pause it whenever you need to, read through the passage, okay? Read through this passage. This is the first question. This is the second question, the third question, the fourth question here, okay? So let's go back. Let's do this. Let's break this down. Write down your answers and see, what you got right, what you got wrong. Okay, let's go. Ovarian cancer is a highly lethal gynecological malignancy affecting more than 22,000 women annually in the United States alone. Although most patients initially show a complete clinical response to surgery and chemotherapy, recurrence and progressive disease eventually occur in the majority of patients. All right, I'm not going to highlight anything here. They give me numerical value, 22,000 women. That's okay. That's not really that important to answering questions. And then they told us that reoccurrence eventually occurs in the majority of patients. Okay. Nothing to highlight really there. The sensitivity of tumors to platinum-based chemotherapeutic agents is a major predictor of patients' prognosis. All right. So these platinum-based chemotherapeutic agents, all right, if the patient's sensitive to a platinum-based chemotherapeutic agent, if the patient is not sensitive to a platinum-based chemotherapeutic agent, that tells us a lot of information on their prognosis, on what happens, the outcome of this patient, okay? Oh, and they tell us, so platinum-resistant tumors are associated with poor outcomes, okay? So if a patient, all right, has a tumor that is platinum resistant, they have a poorer outcome. The disease will probably manifest into something way worse than if the patient did not have a platinum resistant tumor. Okay, it's making sense here. This underscores the need to identify and eventually target pathways involved in platinum resistance. Okay. I don't like how they use the word underscore here. They could use a different word, but they basically want to investigate these platinum-resistant pathways, okay? This task is complicated by the heterogeneity of cells within malignancies. The cancer... Hold on, I gotta sneeze. The cancer stem cell CSC theory proposes that resistant cells encompass only a minority of cells within a cancer, but are solely responsible for long-term reoccurrence. Okay, so it's these cancer stem cells that are responsible for the reoccurrence. Okay, therefore, chemotherapy must target these cancer stem cells in order to show long-term effectiveness. Makes sense, chemotherapy will target these cancer stem cells so that we cannot have the recurrence again. All right. Aldehyde dehydrogenase activity has been shown to be a promising CSC marker in several types of cancers. Okay. Researchers investigated whether ALDH expression in the platinum-resistant ovarian cancer cell line, okay, so this is the resistant cell line, was associated with aggressive tumor behavior. Okay, I'm highlighting things to kind of make a note in my head of what's going on in this research, what's going on here. All right. Researchers investigated whether ALDH expression in the platinum-resistant ovarian cancer cell line was associated with aggressive tumor behavior. Okay, so if I were to put this and rearrange it in my own words, all right, I would say this. I would say that these scientists are trying to find out if ALDH is associated with aggressive tumor behavior. Okay. And it's ALDH specifically the expression in platinum-resistant ovarian cell lines. Okay. If we have high ALDH, is that bad? If we have low ALDH, is that bad? Let's find out. Cells were sorted by whether they expressed high levels of ALDH or low levels of ALDH, and researchers measured invasiveness and response to carboplatin, a platinum-containing therapeutic. All right, and then they gave us a figure here. All right, so we're getting a lot of details coming up here. Okay, we'll have a lot of details here. So when you have a passage that you know is of more difficulty, you want to slow down your reading and making sure you understand sentence by sentence what's going on here. Okay. So they want to measure the invasiveness and the response to a therapeutic. Do we have more invasiveness? Do we have less invasiveness when we use carboplatin? All right. Do we have more invasiveness when we have more ALDH? Do we have more invasiveness when we have less ALDH? You know, they're trying to answer all those questions. Okay. And in the figure, they answer those questions, but we're not going to look at it now. We don't want to waste time. All right. We only look at the figure when the question asks for it. In a subsequent experiment, targeted short hairpin RNA was used to silence ALDH expression. Okay. So after they ran this experiment with the platinum, the carboplatinum, they used shRNA to silence ALDH expression. Invasiveness and percent survival in response to carboplatin treatment were measured in control and ALDH knockdown A2780/CP70 cells. All right. So after the initial experiment, when they used the carboplatin, they then knocked out the ALDH. They knocked down the ALDH. And then they measured, hey, is it more invasive or less invasive when we knock down ALDH? Okay. The expression levels of, actually, I'm going to read this. Don't look at the figure, but you can read the caption here. All right. Invasiveness and platinum resistance in ALDH knockdown cells. Cool. The expression levels of genes in the ALDH knockdown cells were analyzed using polymerase chain reaction. All right, cool. A significant increase, significant guys. They literally told us significant here, so pay attention. All right, that's a key word there. Whenever you see significant, zone in. A significant increase was found in the expression of BAX, a pro-apoptotic gene. All right, when we knock down ALDH, we have an increase in BAX and a significant decrease in CDK4. All right, this is a cyclin-dependent kinase, which encodes a protein that promotes progression from G1 into S. All right, so when we knock down ALDH, we are finding an increase in apoptosis, also a decrease in the cell cycle in proliferation. Okay. Now, this passage was very detailed. Okay, it's definitely one of the harder passages, but it's possible to get them all right. Okay, I'm going to show you guys how to make this easy. Now, question 10. Which of the following is most likely to be a trait of cancer stem cells? Oh, pretty simple here, okay? Ability to differentiate into any cell type in the body. I wouldn't say that that's a trait of cancer stem cells, okay? The only stem cells that can differentiate into any cell type in the body are the totipotent stem cells. And those are found as soon as fertilization happens, right after fertilization happens. Okay, so sperm, egg, they fuse. We have the zygote, and then that zygote undergoes cleavage, becomes more cells really quickly. And those cells there, okay, those can differentiate to anything. They can differentiate into the placenta, and they can differentiate into the embryo human body, okay? And those are only totipotent stem cells, and that's a big deal there, okay? Differentiating to any cell type of the body. So I would say it's not a trait of a cancer stem cell, okay? Like if you have a cancer in the ovaries, that's not going to differentiate into a cancer like of the lungs, let's say, okay? Those cancers in the ovaries differentiate to only ovarian cancer, not lung cancer over there, okay? Hope that makes sense. If it doesn't, comment that down below. That's a big interpretation you have to get there. Okay, so A is wrong. B, self-renewal. Yeah, okay. They told us that these cancer stem cells, okay, they are involved in reoccurrence. So they can self-renew themselves. I like this answer. And that explicitly tells us, that kind of makes sense here of what stem cells are. All right. Greater reliance on aerobic glucose metabolism. They did not tell us anywhere about whether it's better with oxygen or without oxygen. We can't make that assumption, all right? We don't have information for that. Nowhere in the passage did it talk about that. And because of that, we're going to eliminate C. Elevated levels of Bax expression. Bax is a pro-apoptosis gene. So elevated levels of Bax would tell us that we're undergoing more apoptosis. And cancer stem cells, they don't undergo a lot of apoptosis. They go through little levels of apoptosis. They want to keep dividing and be invasive, okay? So this is wrong. All right, the answer is B, through process elimination. And also, this makes sense, self-renewal. Easy. Which of the following conclusions is most strongly supported by the data presented in the passage? Okay. This is a great question because a lot of MCAT questions have this exact wording here. All right, so pay attention to this question. This question is really good. ALDH expression is associated with additional factors that promote invasiveness distinct from platinum resistance. All right, we're going to have to look at the platinum resistance one here. All right, and they're talking about expression. So let's see. Okay, let's look at figure 1 now. This is talking about when they're measuring the invasiveness when we have low to high levels of ALDH. Okay, so when we have high levels of ALDH, what happens when we use carboplatin? Okay, so I could zoom in here, right? Can I zoom in? Okay, cool. Let's see. Dark is little ALDH and light is a lot of ALDH. This is telling me that the carboplatin, all right, in low levels of, okay, so carboplatin works good. All right, carboplatin works good on low levels of ALDH and high levels of ALDH. Okay, it's good at destroying the colonies. It makes the levels less. All right, carboplatin works. That's what this is telling me here. All right, what about the invasiveness? Carboplatin on the invasiveness doesn't really tell me. This just tells me that when we have more ALDH being expressed, we have more invasiveness. All right. Let's look at figure 2 here. This is with the knockdown. So when they knock down the ALDH, that's this one, SHALDH, when they knock it down, I see that the levels of invasion, they're relatively the same. So it doesn't really matter if we knock down ALDH. We're still going to have the same levels of invasion. Let's look at the carboplatin here. Okay, let's look at the controls first. Always look at the controls first. SH control. Okay, we have a lot of survival here. When we add more carboplatin, we have less survival. So good, carboplatin doing its job. Over here, knockdown of SHALDH. We still have some survival, but when we add carboplatin, again, it does its job. So that's good. And let's see, does it job better? Yeah, I would say carboplatin is good. Okay, we need that. So those are my conclusions on this. They're not some crazy conclusions. It's just simple. Is this good? Is this bad? Does this work? Why doesn't it work? Why does this work? All right, it's a very good passage. It's challenging my thinking too, which is good. So this is telling me ALDH expression is associated with additional factors that promote invasiveness distinct from platinum resistance. Yeah, when we look at the invasiveness here, okay, even though we knock down ALDH, we still have invasiveness. So I'm sure there's other factors that we kind of have to test and knock down, all right? It's not solely the knockdown of SHALDH. There's other additional factors that promote the invasiveness here. If we saw it go from this all the way down here, like bar graph was small, then I could say, hey, that SHALDH knocking out ALDH, that is very, very good. That is what we need to knock out. If we knock that out, we're doing really good at targeting this cancer. But that's not what it's telling us. We need additional factors, guys. I hope that kind of makes sense for you guys. I could reword it for you guys again. I think I'll do that just to make it make a little more sense. Okay. If we look at the cells invaded, all right, when we knock down ALDH, we have the same amount of cells invaded. The invasiveness stays the same. So because of that, this means that there's other factors out there that we have to take care of in order to lower the invasiveness. And those other factors we don't know. We have to do other tests for that. Okay, so A is telling me exactly what the data and the figure is telling me. So I like A. B, platinum resistance is the mechanism through which ALDH promotes invasiveness. Okay. We don't know that. We can't say for sure that the reason why there's all this invasiveness of ALDH is because of the platinum resistance. There could be other reasons why the invasiveness of ALDH is so high other than platinum resistance. Maybe it's very good at binding to its targets. Maybe it's very good at not being targeted by drugs. Maybe it's good at diffusing. Maybe it has a specific charge on it that others don't have. Okay, we don't know why ALDH is so good at invasiveness. We can't say that it's so good at invasiveness because of the platinum resistance. We don't have information for that. It's not backed up. There could be many other things. ALDH expression is exclusively responsible for the platinum resistance phenotype. No. Okay. C is wrong. ALDH expression is exclusively responsible for the platinum resistance phenotype. Again, you could have many, many other things. Okay, also, wait. C is wrong. Just because ALDH expression, how is that linked to the phenotype? Like, no, that's just completely wrong. That's two completely different things, guys, okay? D, Bax expression is promoted by ALDH. Bax expression is not promoted by ALDH. ALDH keeps the invasiveness, the proliferation going, all right? Bax is apoptosis. So therefore, Bax, this one's wrong, all right? A lot of wording here, a lot of looking one up, one out. Very, very good questions here, all right? Comment down below if it makes sense for you guys or if it doesn't. I'd like to hear it. Which of the following would be expected as outcomes of ALDH knockdown gene therapy? Okay, let's go back to it, guys. Remember, if we knock it down, there's no difference in invasion. No difference. Okay, I already see reduced invasiveness, so this one's wrong. Whatever has 2 is wrong, okay? There's no reduction. It's the same. Same thing. All right. Slower proliferation. They told us, remember the significant, guys, zone in on that word. Okay, whenever they say significant, you want to read after that very carefully, okay? They told us that their expression of Bax after the knockdown goes up. So we have more apoptosis going on here when we knock down ALDH. And if we have more apoptosis, we're not going to have proliferation. We're going to have slower and slower proliferation. We're going to have more dying out. Okay? Therefore, this one is an expected outcome. This is good. Okay? 3, increased platinum sensitivity. If we look over here, all right, when we knock it down, look what happens. Just simply knocking it down and using the carboplatin, yeah, we get decrease of survival here. All right, the carboplatin works better if we knock down ALDH. Okay, look how much it goes down. And it goes down. All right. So therefore, 1 and 3 are correct, and I'm going to go with B here. All right, very, very good questions. This is section bank difficulty. I would say it's probably a little harder than section bank difficulty. I really like this passage. Let's keep going. If true, which of the following findings would most likely directly challenge the results of this study? Wow, very good question as well. They want us to really understand what this study, they want us to really understand what's going on here. ALDH expression correlates with the expression of other determinants of tumor invasiveness. Okay, this is wrong. That's wrong. Okay, how does that challenge the results of this study? What do I care about other determinants and how they correlate with it? That doesn't tell me anything. Okay. Targeting a different pathway instead of ALDH yields better improvements in platinum sensitivity. That would just tell me information like, cool, like, thank you. Not like thank you, but like, this would just tell me, okay, cool story, bro. Like, we don't care about the other pathway. We care about this pathway. All right, this pathway is what matters, not a different pathway. Okay. No significant relationship is found between ALDH expression levels and clinical behavior in non-carboplatin-resistant ovarian cancer cell lines. Okay. Why do we care about non-carboplatin-resistant ovarian cell lines? This whole thing is about the platinum-based resistance. Okay. The platinum-resistant ovarian cell lines. Okay. We're talking about resistant, not the non-resistant. All we care about is the resistant. All right. So this one you can eliminate here. Don't be fooled. Don't be tricked up by the wording here. Non-carboplatin resistant. Don't be tripped up by that. Okay. Stick to what you know and stick to the evidence you see. Passage talked about platinum-resistant ovarian cell lines. Passage did not talk about non-carboplatin resistant ovarian cell lines. And because passage did not talk about non-carboplatin resistant ovarian cell lines, C is wrong. Okay. D, ALDH overexpression results in no significant differences in platinum resistance compared to control cells. Yes, this would directly challenge it because remember here, okay, the whole data here said, hey, when we have more ALDH, okay, when their expression is higher, we have a difference in the resistance. Okay, look, here's ALDH and here's with carboplatin. Okay, there's a difference there. We talk about the expression here. So D is the best answer. Also process elimination, guys. If you really didn't understand this, you could do process elimination and kind of get to B as well. All right, let me know in the comments if this makes sense, okay? And that's it for this passage, okay? Very, very good passage, guys. If you want me to explain things a little further, just comment it down below. I will, okay? I won't, like, don't hesitate to explain it. Like, don't hesitate to comment down below something and ask me to explain it further. Okay, this was more than section bank difficulty. This is a pretty hard passage. MCAT is easy. I like to say that a lot, but some passages, they're meant to trip you up, like this one here, okay? Comment down below what you guys think, and I'll see you guys in the next video.